Okay, in 7.4, we are looking at the idea of pedigrees and things like that with human genetics and seeing how those work. Human genetics follows patterns seen in other organisms. Basic principles of genetics are all the same in all sexually reproducing organisms. Inheritance of many human traits is complex, though. Single gene traits are important to understanding human genetics. Females can carry sex-linked genetic disorders and not show them. Once again, males, which are XY, express all of their sex-linked genes, ones that they inherit from both mom and dad. So that's why they have a greater chance of showing a sex-linked disorders because whatever they inherit, once again, they're going to display. So expressions of disorders depend on which parent carries the allele and the sex of the child. So if we look at these X chromosomes that are at the sex chromosomes that are there at the bottom, you can see some known examples of sex-linked disorders. So Duchenne's muscular dystrophy, the retinitis pigmentosa, X-linked deafness, Fragile X syndrome, red-green color blindness, hemophilia, testes determining factor, testes specific transcript. So those could be errors that could show up on any one of the sex chromosomes for the guys. So those are some common things. A pedigree is a chart for tracing a gene in a family. So phenotypes are used to infer genotypes on a pedigree. Autosomal genes show a different pattern than what you would find with sex-linked genes. So right here is one that's showing a autosomal one. So you can see it's an equal opportunity one. If we're looking at it, females are circles, males are squares. If the person has the trait, it's going to be filled in, or if you know they're a carrier for it, it's going to be half shaded. So this is an equal opportunity one. You see both males and females equally with the trait. Next, if the phenotype again is more common in males, the gene is most likely a sex-linked trait. So you can see in this pedigree that the mom was a carrier. She had two boys that had the trait, and you can see further down that the one oldest daughter had a son that had whatever trait we were looking at here. So several methods help map human chromosomes. You can use what's known as a karyotype, and that is a picture of all the chromosomes in a cell. So they just basically take a picture of your chromosomes, and they're going to cut them out and put them with one another. Karyotypes can show changes in chromosomes. So it can show things like deletion of part of the chromosome or a loss of a chromosome. It can show large changes in the chromosome, so like an extra or a duplication of the chromosomes. So right in this one, you can see that there are several that you have different errors with the chromosomes there. So that would be a karyotype right there that you would be able to see. So we will be doing a karyotype lab in class. So that is the end of the notes for section 4, and it's also the end of the notes for chapter 7.